I'm going to start in what I hope will be a useful juxtaposition of the past and the future, which I hope in some ways will set up the rest of this fantastic program. And I want to reflect a little bit on what we've achieved in the last decade or so. And I'm going to focus my remarks on rheumatoid because it seemed coherent and cogent with what Mike and others are going to talk about later. And then I am indeed going to show and share with you some data from the lab, just to give you a flavour of the kind of work that we are doing, and it's not that it's any better than anyone else's work, but it happens to be data we've generated. And most of what I'm going to show you is unpublished, so you're amongst the first to see it. So if you think it's rubbish, tell me, because it'd be good to know before it goes away to the editors. And then I'm going to take you back into the clinic and show you some of the work that we are doing, taking those very same molecular understandings from the lab into the clinic. And I'm going to talk a little bit about the next generational challenges. So I hope in all of that there'll be a little something that will be interesting to you. I'm going to start though with heparin. So in the United Kingdom, for as much longer as the United Kingdom lasts, I'm from Scotland right now. My fingernails are slightly broken because I've been hanging on to the border of England very tightly for the last 2 or 3 years, and now we're hanging on to Europe very, very tightly. But I think if you come to visit us anytime soon, you'll need to get the pilot to land somewhere mid-Atlantic. We'll be floating listlessly, waiting for something to happen. But, you know, in the United Kingdom, we have our own little historians as well, Glenn, and I thought I'd start with Heberden. And many of you have maybe heard of this story before, but Heberden was a remarkable physician with a very great interest, amongst other things, in musculoskeletal disease. And Heberden was also a scholar. When we think of it, it was extraordinary listening to the range of achievements of the Cleveland Clinic physicians. I actually think as physicians and as health professionals, most of us have other interests. I think that's why we are physicians and health professionals, because it's more than a science, isn't it? It's an art, looking after people. And I'm going to come back to that at the very end of my lecture today, with what for me was a disappointment and then a pleasure when I thought about it in the final experiment I'll describe. But Heberden published The History and Cure of Disease as one of the great historical medical texts. It was published the year after his death and was actually under the auspices of his son, and I've always thought that was a great act of love on the part of a son to ensure that his father's life's works were published, even though he was departed. And Heberden was a great believer in asking questions. He saw no difference in clinical practice in research, well, we now call it clinical and translational research, but actually to the physicians of the past, asking questions about our diseases, being curious, was simply part of standard of care. Would that we could go back to those wonderful days in these very metricalized and challenging and electronic health delivery lives. And when you go back into the origin of the word, care is very interesting. Curiosity and care come from the same Latin derivation. The actual word care means curious and vice versa. So let me start by reflecting a little bit on what brought us here. Now, I'm very aware that for some of you it's early in the morning. For those of you from California, it's also very cold. So I'm going to give you the things that you need to know in the next 10 minutes in one box. So this is for CME. So what I'm about to tell you is in this little blue box. I'm going to tell you that we've had an amazing decade or so in the treatment of rheumatoid, but you all knew that. I'm then going to be just a little bit, and forgive us, Scott, a little cynicism. I'm going to take a slightly dry view of that progress. I'm going to think a little bit about what we've learned about the taxonomy of immune-mediated diseases. And I'll come back to that at the end, but I think in 10 years, disciplines will have dissolved as we take the molecular taxonomy of our immune-mediated disorders forward. And I'm going to then remind you about the evolutionary imperative to have an immune system and why I think we need to revisit that in designing the therapeutics of the future. And then that, I hope, will inform how we deal with one of the biggest challenges in rheumatoid, which is refractory disease. And I've chosen that because with Mike talking about pre-RA and early RA, I hoped that dealing with the chronic and refractory element, in a way we're almost talking the wrong way round, but I'm not brave enough to talk after Mike, and my tie wouldn't stand it either. So I was very lucky I had a tie today. I've never seen you in a tie, Professor Len. It's actually quite extraordinary, a very joyful moment for me. So I think a few of the basics. Everyone in the room knows that rheumatoid is a systemic inflammatory disease, and I hope everyone in the room is also very aware that it is a disease of multiple comorbidities. It's a disease of the entirety of the person. And increasingly, when we're thinking about pathophysiology, we should be thinking not just about the synovium, although that will be my focus today. We should be thinking about why that inflammation, which has at one point a synovial impact, is also manifest in the major blood vessels of the periphery and the brain. Also in the metabolic beds, adipose tissue and rheumatoid is an unhappy place. So too systemic bone, and so on and so forth. And the facts are that our patients die, and they die earlier than they should as a result of having rheumatoid arthritis. Mortality is probably improving at the moment, and I won't use some of the more hysterical numbers that are sometimes put about when we look at historical cohorts. But nevertheless, our patients are more likely to succumb through vascular, infectious, or oncologic reasons, and they'll do that earlier than they otherwise should have. And that's a terrible waste of life, particularly when aggressive management and understanding could prevent it. But the earlier recognition that rheumatoid was a disaster, functionally, and in terms of morbidity and survival, when you put that against our therapeutic responses to it up until around the year 2000, it's pretty sobering. We were using non-steroidal drugs, willowbart derivatives, glucocorticoids, remarkable and noxious at the same time, particularly if they're used in more than a dose of around 8 milligrams a day. And then the advent of conventional DMARDs, Nana Schwarz's work in the 40s, sulfasalazine. That's SULF or PHA, just for my American friends. It's a drug we use a lot in Europe, but only in some parts of the United States. But actually we use it particularly in our combinatorial regimes, as you well know. And then the really remarkable advent of biologic therapies, which are celebrated in this series of summits. I've started with anti-TNFs because that was the beginning, but it could have been any of the cytokine inhibitors that we now see, because the evidence in favor or against their use in rheumatoid is pretty much common across the drugs. There was very little difference when you compared the biology as then known of IL-1 and TNF, and yet the way those targeted drugs have gone in rheumatic disease practice has been very distinct indeed. And that, of course, is Ehrlich in the corner of his office. This is in the laptop days, and I have this notion in my head that he knew where every single paper was. I bet he did, and if any of you have the chance, you can go to Charity in Berlin and go to the university, and they have his office still preserved, although it's a bit tidier than it was then. So what did we learn from biologic therapeutics? Well, I think the major celebration was that people got better. So we've rightly celebrated and focused on the clinical improvements. We've rightly focused on reduced morbidity and mortality. I think we've paid a lot less attention to what this has taught us about the immune system. And I regret that. And I particularly regret that we haven't paid attention to the drug failures as we have developed our understanding and idea of the rheumatic immune system. So we learned that TNF blockers were successful, IL-6 receptor blockers, and we now know 6 ligand inhibitors were also successful. We learned that IL-1 blockade was probably not terribly useful. It does work in some patients. All of us probably have the odd patient who did okay in anakinra when we prescribed it. But it wasn't a dominant pathway. And then we went on with the advent of abatacept, rituximab therapeutics, to understand that you could also move upstream of the cytokine networks into co-stimulation, linking myeloid cells, dendritic cells, macrophages, T cells, and doing something to the B-cell compartment, a subtle approach, wiping out a large proportion of your CD20 cells, which are pre-B cells through to plasmablasts, but not touching, rather interestingly, the plasma cell. And we just kind of went on. We've had these drugs now for more than a decade. Do we know what order to use these drugs in? Do we know what drugs should be used first? What is the plausible combination? What is the appropriate patient to receive one, the other, or the next? What have we been doing with ourselves? Have we been away celebrating on the golf course? I never celebrate in the golf course for the avoidance of doubt. I play with my daughter and it costs me a large amount of money on an hourly basis, let me tell you. And now with the JAK inhibitors, we've learned that you can move inside the cell and you can inhibit immune receptor signaling and achieve remarkably similar clinical outcomes, but we've not easily broken through the ceiling. The only things that have really taken us through the ceiling have been strategic interventions. So actually, ladies and gentlemen, one of the major lessons of the last decade is that I think we're really bad at choosing targets. That may be ironic when we've had success, but actually we've had so many failures that if we celebrate those rare successes without reflecting carefully on what did not work, and that's a very Scottish view of the world, I apologise for it, but I think there's a lot of evidence to support it. So how do we actually choose the right targets? Which are the most important targets that will capture the response? So the difference between a TNF and IL-1 blocker is that when you block TNF, the whole response collapses down, so people get better. When you block IL-1, that doesn't happen. Note well, it does not mean that IL-1 is not bioactive in the joint and doing bad things. You'll often hear people being rather lazy and saying, IL-1 is not important in rheumatoid. Well, I rather suspect IL-1 is important in rheumatoid. There's lots of it there and it's activating chondrocytes and osteoclasts and fibroblasts and macrophages, and those cells are doing bad things. It's like saying that the guy who plays with Cristiano Ronaldo for Real Madrid isn't actually terribly important. This is complete nonsense. Ronaldo, I think, believes that may be true, actually, but no one else. So how do we make the right target? Think of Scotland for a moment. This is Scotland, still attached to England, you can see. And here's Glasgow and Edinburgh. Now, some of you, if you take a very superficial look at Scotland, may think that Edinburgh is this beautiful city with a castle. And actually, when you really think about the culture and nature of the great cities of Glasgow and Edinburgh, if you are interested in a place with intellect, creativity, hard-working spirit, and a great sense of humour, then you must never, ever, under any circumstances, go to Edinburgh, for you will be sorely disappointed. I can't really help you other than with the following little anecdote. If you get in a bus in Edinburgh, and there are only two of you there, plus the driver, one of them will go upstairs and the other will go downstairs. In Glasgow, in an empty bus, two of you will be sitting next to each other. They'll offer you a chip. That's a French fry out of a piece of paper, little heavily double fried, the Glasgow variety. And by the time you get to your destination, you will be bosom buddies and friends for life. And if you're very unlucky, you'll know their entire history. And if you're from America, they'll tell you they're related to you as well. So we aren't very good at choosing targets, and I'll show you how I think we can deal with that later. The other thing we've learned from biologics is that all diseases are not created equal. The original idea that TNF was the super cytokine, the master regulator, is simply probably untrue. Certainly the idea that IL-6 and TNF sit next to each other as partners and twins is absolutely untrue, and the spondyloarthropathy and giant cell arteritis and rheumatoid clinical trials have shown that. And all we're depicting in this little cartoon is the idea that different disease states have defined responsiveness to different biologics. And that now we know about the inflammasome, we understand why IL-1 blockade works in diseases like gout and JIA. The inflammasome is the molecular machinery that processes the IL-1 family and allows neutrophils and macrophages to behave in a very inflammatory way. And we're updating this cartoon, it's already out of date. But a quick word for strategy. Strategy, depending on how you like to see the world, may well have started, amongst others, with the Cohort study in Glasgow. Simple competition between the doctors and the nurses. Doctors treated the patients because they were brilliant. The nurses treated the patients and measured a disease activity score. The nurses got 2/3 of patients to remission. The doctors didn't. We were 16%. So that was a resounding victory for the Glasgow nurses. Cohort study was then published, and I think most of us now accept the concept that we will have some kind of target. And interestingly, when you look at the outcomes for Cohort, they're still at least as good as any biologic therapy until very recently. And that was no biologic use at all. Clearly showing us that the right use of a drug is going to be every bit as important as the right target that the drug takes out. And I think from Artie and others, we're going to learn a bit about tapering, which is the next step, is going to step 1 and then coming back from step 2. And yet, ladies and gentlemen, I believe one of my predecessors in this lecture was Professor Joseph Smolin. This is the Smolin tartan. I'm having a kilt made for Joseph for this summer so that he can wear these colours. And it's a beautiful cartoon that he and Daniel wrote for a review a year or so ago, in which he moves from left to right through methotrexate naive to TNF-experienced patients and depicts in green high proportion of responders and in red low proportion of responders. And the facts are that as patients progress through therapeutics, their magnitude and quality and depth of response reduces. Why? I didn't hear any answers there, so I better move on. I have a proposal for you momentarily. This is actually the primary question that's driving my lab, and I'm going to show you what we're doing to trying to get to that. And then the other lesson that I think we've forgotten, and no better place than a clinical immunology centre to remind us, is that the immune system was not generated for the gratification of pharmacology, rheumatology, and pharmaceutical investigators. It was, of course, invented to defend us. This actually is me in Malawi, and over dinner, if you're interested, I'll bore you to death about what we're doing in Malawi, but we're integrating an infection and rheumatic disease program to try and understand the common pathways that define infectious and autoimmune-mediated pathways upon human health and outcomes as part of many, many efforts around the world to understand non-communicable disease. But the reason I highlight this is because, in fact, if we think about chronic autoimmune disease as an aberrant infectious process, then suddenly a lot of things make a lot of sense. We don't generally think about it in those terms. So, the first thing is that everyone has an immune system right back through evolution. And forgive me, one or two of you know what's coming, but this is the Scottish football supporter. This is the highest evolutionary form of mammal. It is resistant to all manner of disappointment. There is nothing that our soccer team can do that will allow us to become unhappy. And they've tried very, very hard. Our 96th minute victory against the mighty Liechtenstein under-11 girls team springs to mind. We have an immune system to defend us. We have evolved with a highly dynamic immune system. Think of the work of Max Cooper and others. The trouble is the bugs are smart. So this, everyone remembers, is an African trypanosome, and trips change their surface antigen coat. They put a new jacket on every few hours to make it difficult for the immune system. So the immune system, I use this as a single example of the common truth, which is that the immune system needs to know where it is, when it's there, what it has to do while it's there, and it has to deal with a dynamic changing host. So ladies and gentlemen, when patients come into the clinic with rheumatoid arthritis, do you really think their immune system will be the same 6 months, 1 year, 10, 20 years later? This is not evolutionarily sensible. And yet we treat rheumatoid as if the pathogenesis, singular, of rheumatoid is as follows. And what I hope you'll see when Mike speaks and some of the data I'm going to show you in a moment is that actually the immune system is dynamic, I believe, in the disease, and it is our failure to recognize that has led to so many therapeutic disappointments. We don't really think about the compartmentalized, we think about the synovium and the blood, and we argue about that, but we don't think about the kinetic compartments of the disease. And I believe this is costing us clinical responses. So here's a way of thinking about it. You could have cytokines that are involved in pre-disease, disease onset, active disease, and those may define people who will not respond, A, or will respond, B. You may have other cytokines, immune factors, that will regulate maintenance of remission or failure of remission. Part 1 is over. If you're still with me, you can now sleep for about 10 minutes because this is the immunological bit. Professor Calabresi. I anticipated that this may be a little demanding at this time in the morning as well for some who are very, very clinical, and for that reason, here are the key learning points of part 2. I'm going to suggest to you in cartoon form that pre and established RA are probably different. I'm going to suggest to you that if we understood those pathways that define the differences, that would lead us to plausible approaches to refractory disease. And I'm going to put the macrophage rather than the T cell, which as a T-cell biologist for many years is kind of interesting. But we turned to macrophages a few years ago, and I'm going to show you some science we've done in the macrophage. So here's the current model of the disease in ridiculously simple form, for which I do not apologize. We have a pre-arthritis phase that we're going to hear all about from Mike, which is a kind of environmental, genetic, epigenetic interface. That leads to some kind of breach of tolerance. My own view is that rheumatoid is a failure to understand and regulate the maintenance of tolerance to post-translational modification, when proteins are changed and modified, because there's lots actually of antigen specificities coming through, and this makes one very suspicious of a single antigen solution. And then that eventually causes this swelling on the right-hand side. Now, it's very well known that radiographic progression relates very closely to functional loss. It's also very well known that if you delay the onset of treatment, radiographic progression will be greater. What is less often appreciated but is very consistently detected is that the slope of progression is greater. That is, if you start off with more damage, you will accelerate the damage process at a greater rate. Not exponentially, but nevertheless, greater rate. So how does the body sense damage? The body senses damage through a family of large family of molecules called the toll-like receptors. And these are the part of the body's damage sensing, and they're designed to sense bugs, microbes, and they're smart. There's got to be quite a few of them, and they actually are dynamically regulated. And remember the trypanosome flying through the cerebral artery into the brain. Think about that dynamic, incredibly agile immune defense that is necessary for that evolutionary imperative of the immune system. And what I'd like you to think about in terms of chronic rheumatoid, and this is very different from the early phases, we can have a discussion later about how this works in early disease, is a vicious circle of tissue, osteoclast, chondrocyte, fibroblast, and innate immune, mainly macrophage biology, driving a damage response. So here's a macrophage, and I'm slightly... sometimes you put a figure together. This figure we put together for an article Gary Firestein and I just published in Immunity, and this is about 15 years of my life in one slide, which is, and actually we just recently had a retirement of my senior technician. Jim worked for the University of Glasgow for 48 years. And we actually brought photographs of the lab together from the last 20, and there's a lot of people have done this work, and I've done almost none of it. But we've, over the years, worked out that macrophages are activated by integrin interactions with T cells. Actually, Tiny Maney's group and others worked out that immune complexes, and this has been beautifully developed by Jeremy Sokolov and others who have really helped us understand how the FC receptor and the toll-like receptor families work together. The late Brian Foxwell was one of the first to describe that. Sensing damage. We previously showed that proteases through the PAR2 receptor system, macrophages sense the amount of enzyme around them and are activated by that. We've worked out that oxysterols drive macrophage activation through the LXR nuclear receptor. Oxysterols, ladies and gentlemen, are derivatives of dysregulated cholesterol metabolism. Does that ring a bell? Rheumatoid is associated with dysregulated cholesterol metabolism. And actually, there's some very, very provocative epidemiologic data that we put together with some of the guys over in Harvard. So the metabolic dysfunction is evident in pre-RA. And it's even possible that dysregulated cholesterol metabolism is a pro-inflammatory factor as well as a consequence of inflammation. The directionality in the lab is often very different from that we assume in the clinic from epidemiologic studies. And very recently with Gordon Brown up in Aberdeen, we detected novel autoantibodies against inhibitory receptors in macrophages, so-called anti-MICL. I won't dwell on the details today, but a really interesting interaction between autoantibody that led to a dysregulation of the macrophage, the adaptive response dysregulating the innate response, which would be a recipe for chronicity. So the question I want to tell you about in the next few minutes is, what is the master regulator of all of this? How are all of these exogenous signals into a macrophage integrated? And could that give us a clue either to new targets, or could the master regulators inside cells lead us to new pathways? So, this is C. elegans, it's a worm. This is a petunia, I'm reliably told by my wife. They are commonly linked by regulation of their gene transcription by microRNA. MicroRNA is exactly what it sounds. It's an RNA, but it's only 22 base pairs long. And the way it works is that the microRNA binds to the bottom end of a normal RNA and kills it. It codes it for destruction. So, for the next 5 minutes, I need you to think the wrong way around, ladies and gentlemen. If you see a microRNA going up, its target goes down. Everything's going to be the wrong way around. I just came from Scotland to London to Chicago to Cleveland, so everything's upside down for me today anyway, so go easy. Right. So we went looking for microRNAs in synovial myeloid cells, and we found lots of them. Well, actually not so many. There are about 1,200 microRNAs described in the mammalian genome, higher mammals anyway, even Scots. And we found several dozen of these were alternately regulated in the synovium, and we chose first of all to look at microRNA 155. And the lovely thing about a microRNA, if you think about it, is you know its sequence and you know its target sequence, so you can ask the computer to say, which genes does this microRNA regulate? And one microRNA usually regulates 10 or 20 different RNAs. So this is a bit like the coordinator of FedEx sitting in the office saying, I need 20 lorries at Cleveland Airport, 2 o'clock this afternoon. But to do that, he needs to control fuel, drivers, and trucks. Sorry, lorry, truck. I got a blank look from Mike there. So you got to coordinate all these. Have you ever wondered how the cell gets all the right proteins into the right place at the right time? I worry about that sort of thing. I have a very sad life. MicroRNAs are the ones that do that. So we then looked at all the RNA in these cells, and this is the RNAs that are up or down regulated in synovium compared to blood. We put them together and we found that 155 was a really interesting molecule. It targeted lots of really interesting pathways in the cell. This work we published a long time ago, 5 years ago. So the yellow cells are macrophages in a synovial biopsy that have the 155 in it. And when we put 155 into a blood monocyte, TNF poured out, especially when we added a damage molecule, which we use LPS in the lab. But for LPS, read probably citrullinated fibronectin would be a good example in the rheumatoid example. So think of that as a damage-sensed pathway. And micron 155, this is not a subtle increase. This produces industrial amounts of TNF and IL-6. And when we knocked this out in a mouse, the mouse in the red got less arthritis. We then went looking in the blood of patients, and this is work that we just published very recently, actually. This work I'm really proud of. It's done by Aziza El-Mazmari, a Libyan rheumatologist who did much of this work while her family were being bombed in Benghazi. This is just an extraordinary bit of work for someone to have the focus and presence to continue to think about these issues in that terrible conflict that her family found herself in. So this is, for my lab, a very special paper, actually. And Aziza developed a new assay, not just to measure the relative amount of these microRNAs, but actually to count the actual number of microRNAs in the cell, the so-called copy number. And when she did that, she found that there was a relationship between, in circulating monocytes in people, the actual amount of micron 155 with the disease activity score in patients. And this was especially so when we looked in patients with ACPA-positive disease. So we made an anti-155 and a so-called antagomere. This is not a facile effort, by the way. This took some time. And here's mice developing arthritis, and here's the antagomere-treated guys in the bottom who developed much less arthritis. So life was great, and because I'm interested in comorbidity and metabolic disease, we did exactly the same experiment in the lung and in the joint, and we were getting excited about 155 as a target, and this is what we found. The 155 knockout mouse, given a little bit of bleomycin, which causes some fibrosis in the lung, this deep blue lung is a very, very fibrotic lung. And then we started to treat some of our 155 knockout mice with a Glasgow diet, a high-fat diet. And this in the bottom right-hand corner is their liver. And these bright orange blobs are cholesterol deposits. It turns out, ladies and gentlemen, that 155 is a thermostat, an immunostat. In an inflammatory environment, it drives cytokine release. In a metabolic or other challenged environment, it actually attempts to repair tissue. And therefore, if we block 155, we're highly likely to develop hepatosteatosis and an increased propensity for pulmonary fibrosis. And so, as Robert Burns would say, the best laid plans of mice and men gang after glee. In other words, think again. He actually wrote to a moose in the old Alloway Kirk down in Ayrshire. And he wrote it in church one Sunday morning. And he was sitting, he wrote to a loose and to a moose. I'll need to translate. To a mouse, to a louse, little insect thing. And the mice were running around the back of the church, and the louse he took very gently out of the hair of the lady sitting in front of him, who was an attractive young woman. It is said that they had a very fine afternoon together thereafter. Burns was a bit that way inclined. He was a terrible man. Wrote some of the greatest poetry on earth. So this is a summary of this phase of what I want to talk to you about, and the summary is as follows, that the synovium in inflammation has told us much, but the synovium in remission may yet tell us more. And that, I think, will lead to pathways that can help us to understand refractory disease. And finally, in my last few minutes, I want to turn to people. And this is, in a way, I'm very grateful to Jonathan, who's going to talk about biomarkers this afternoon. But this, again, for those who really by now must be desperately needing sleep, this is what I'm going to say in these last 7 or 8 minutes. I'm going to say that biomarkers are, at the moment, not a reality in our disease beyond maybe ACPA and CRP, but let's not argue about the fripperies. They don't really allow us to make the highly specific decisions we seek for the precision medicine generation. I'm going to introduce the polyome to you. You'll hear a lot more about that as the days go by. And I'm going to talk to you about the integration of the polyomic data, these molecular and cellular signals in patients where maths meets medicine. So precision medicine, if any of you are interested in the field, this is a fantastic report the Wish Foundation published a few months ago, in which it predicts a world in which molecules and molecular precision medicine approaches will actually define the whole of medicine. I rather hope it doesn't. I hope I can talk to people in my clinic in a few years' time, but this is one extreme view of the universe. The polyome is simply the sequencing of the genome, the transcriptome, the lipidome, if you like, the metabolome, characterizing the structure of cells, and then all you do is put it all together. And this is the problem. Some of you with children will empathize with Wilmot's quote. I have only 2 girls. I have 0 theories left. I brought one of them with me to Cleveland to see if it would help it out. But yeah, 8 hours in a plane from Heathrow to Chicago, and I'm no wiser. The problem here is putting the whole thing together. And it's actually been an informatic challenge rather than a sequencing challenge. We can sequence everything you can look at the moment. But it probably matters. So the final omic is economic. Apologies for the pun, but it's really important. And this is, Victor Zal published this in Lancet. I'm worried and disappointed that rheumatic musculoskeletal diseases are not up here, but you can see that if you could actually reduce the incidence of these diseases by appropriate prediction and prevention, and Mike's going to come to this just in a moment, the cost savings to the healthcare economy are simply remarkable. You don't need to predict 100% of everything. You just need to go after increments. And it's a little unfair that the biomarker field has been hopelessly compromised by the demand that we make absolute decisions with molecules that we've never demanded of new therapies. We need to relax the rules a little. So I guess you've all had this clinic where you're not quite sure what's going on in the patient because they look the same to you, but what they're thinking inside might be very different indeed. We don't know what biomarkers to measure. We don't know whether to measure them in blood or synovium. Should we be looking at DNA? Should we be looking at metabolites, biochemistry? So to address this, here's SERA1. You're going to hear SERA. Well, we can argue about whether it's SERA1 or SERA2. There's another SERA up here in this wonderful part of North America. But the SERA that we invented was the Scottish early RA cohort. The Tartan Cohort. We'll maybe give it a different name just so there's no confusion. You'll see why I made that remark in a few minutes. So we gathered around 1,200 patients. Every rheumatologist in the country contributes to patients. They don't get paid anything. They just do this because they're really committed. And we, in the end, you don't get every patient agreeing to come into the cohort, but we have about 2/3 of all rheumatoid that came to Scotland in that period of time. And we basically analysed the wazoo out of them. We took as much of them out that we could. We have about 80,000-90,000 samples in the biobank. Everyone in Scotland has a unique healthcare identifier, the so-called CHI number, and that means that's all uploaded automatically. So every time a patient in Scotland goes to the pharmacist, goes to the ER, goes to the dentist, gets a drug, it's all uploaded automatically. We then access that through electronic linkage. Actually, we can do that in perpetuity. For a small cohort of patients called the Nested Progression Study, SNAP, we took more than 50 new RA patients and we immunophenotyped them looking at 488 different white cell subsets. So let me show you 3 experiments we've done before I finish. The first experiment, can epigenetics help? So epigenetics, for those of you not aware, is the molecular changes that are enacted on the genome that change the behavior of the genome. Maybe the addition of a methyl group or an acetyl group. And that's because each gene, you vaguely recall, has exons and introns, and it has a promoter. Now, when I was at medical school, there was a promoter. Sadly, nowadays, we've discovered that any gene usually can have several promoters, sometimes 15, 20 promoters. And the genome organizes that by bringing these things together. And if you can think, imagine, I'm about to break somebody's piece of paper. It's my CV, so I'm allowed to do that. Imagine that's the genome, and I want to bring a promoter, which is my left hand, together with the first exon, which is my right hand. The way it does that is this. It loops. Okay? Got the loop. So, clever thing, if my fingers come together now, a new sequence is formed between my left finger and my right finger. I tried to explain that to the man at immigration yesterday. When I was getting the right hand up, he said, "Your other hand, sir. It is my right hand." We got there in the end. The computer was down, he said. So we just measure which loop came together. And we do that, and we did that for about 13,000 different loops that come together. And this is a bit like creating, this is an ordnance survey map. That's actually Loch Lomond, okay? This is Loch Lomond there. And what this ordnance survey map does is it tells you how high or low the ground is around the loch. So it doesn't just tell you that Loch Lomond's there. It doesn't tell you the mountain's there. It tells you how big the mountain is. Is it a lot of work? Is it steep? Is it hard work? Or is it an easy walk? That's what the epigenetics tell us. They tell us not what gene is switched on, it tells us how it's switched on. And we went and looked at the GWAS targets of rheumatoid, and then we looked at another few genes I was interested in, and we looked at the loops, and here they are. We looked at responsiveness to methotrexate in the SERA cohort. 5 genes separated responders and non-responders. I can show you what the genes are without any fear of replication, trust me, because the information here is not that those are the genes, it is how those genes are switched on that is really critical. And it turns out to map, for the geneticists in the room, to EQTLs, to the expression loci that drive rheumatoid. And when we put that into our model, we can pretty much choose 90% of the patients who will not respond to methotrexate. We've now run this through 3 cohorts, and it's standing up pretty well in terms of that number. We're hovering around 88 to 90%. So we might even publish this eventually because we wanted to validate before we published. There's so many unvalidated markers out there that are published. Second experiment, do the cells in blood. Now, I'm a big believer in blood biomarkers. Synovium's for investigation. Synovial biopsy is not for routine clinical decision making in anything other than an extreme scenario in my view. So we've marked 488 phosphostats, ZAP70. Think of a really odd name, and there's usually a protein that's been given that name. So we then took these cells out of the blood, we fixed the cells immediately, or we activated them for 14 minutes, 15 minutes, or 16 minutes. No longer, no less. And we found, for example, that in early RA, CD4 T cells and CD8 T cells have an abnormal STAT3 phosphorylation signal. But there was no signal in circulating CD19 B cells. We then went and looked at those numbers and looked at those patients who were responders to methotrexate and those who weren't. And on the left, you can see that there's a relationship. CBL is another signal pathway that's phosphorylated in the T cell receptor. This, by the way, is complex immune informatic analysis. This is not just looking at a cell, so there's mostly maths. And we were able to see that actually in those patients who are capable of responding to methotrexate, there's a linear relationship between the way the STAT3 pathway is switched on, and just to keep the clinical juices flowing, those are the pathways that the JAK inhibitors go after. So the non-responders don't have that relationship at all. They probably have an immunologically different endotype, a different type of disease. And finally, can we apply this in the clinical trial world? Here's TNF blockade versus rituximab, head-to-head study. We did this, the ORBIT trial, published it last year. And we found, probably no major surprise, that B-cell depletion and TNF blockade were pretty much non-inferior. If anything, it favored rituximab. Duncan Porter led the study. Duncan's Scottish. Well, he's actually originally English, but now Scottish, and he counts the pennies very carefully as a result. And it's quite a bit more cost-effective to use rituximab than it is to use TNF blockade. So you'd like to know who was going to need the TNF blocker. So we've now RNA sequenced the blood of all of the patients in the trial and generated immunologic gene signatures for those most likely to respond to rituximab or TNF blockade. And you can see we're hovering again around the 90% in the ROC curve. So at the moment we can now probably, and these are all baseline signatures, this is a baseline signal that predicts likelihood to response of a B-cell depleter or a TNF blocker. And they're different signals. They're selecting different kinds of patients, which is what you kind of would expect. So if we're going to make a difference, ladies and gentlemen, if I may close with a few remarks, we're going to have to do it differently. You know, the old, the leopard, if you want things to stay as they are, if we want this amazing progress, we're going to have to change. We're going to have to embrace the principles of systems medicine. We need to bring maths and medicine together. We need to take biology, imagination, and in silico discovery and put them into the same space, and it's really hard. I started thinking about this about 5, 6, 7 years ago, and we're still really struggling to put it together. We don't have the resources of the Broad or Hood and the guys in Seattle, but we're trying very hard. So here's a synovial tissue. So now we wouldn't think about the linear TNF IL-6, IL-1 pathway that was the brilliant hypothesis that brought us TNF blockade, and not for 1 minute am I disputing that. It was extraordinary. But that's not what we really see. The problem with a single pathway, this is the Glasgow Underground. It goes this way or this way. Now, it rains a lot in Glasgow. I don't know if I mentioned that. And it floods in Cowcaddens. And if the railway floods in Cowcaddens, you can go neither way. The whole system shuts down. A single linear immune system will not survive the microbes. Hence the emphasis on evolution. So it has to be a complex system. And there are many cytokines, both pro and anti-inflammatory, described in the joint, and here are just a selection. So think about them in terms of host defense. The collection of cytokines designed to work together for gram-negative defense are IL-1, TNF, IL-6, GM-CSF, and these are the molecules that nature defined would be optimal for a gram-negative infection response. That's the perfect team for that game. For rheumatoid, it's not that team. Some of those team members can contribute, but others are not necessary. They are contributing, but not functionally essential. There is another way of thinking that it's regulatory redundancy. So think of your soccer team again. Jose Mourinho, Manchester United. He would pick his best team to play Chelsea, but he would probably need a very different team to play my team, Glasgow Rangers, who are awful. So the psoriasis network is 17, 23 TNF. IL-6 doesn't need to be involved. This is the way we have to think about it. That's the easy bit, unfortunately, ladies and gentlemen, because the parameters that surround the immune system are so much more complex. You can just cast your eye around all of the elements here. Look to the left-hand side of this slide. Cognition, the environment, emotional status. We know that emotion and depression and anxiety and life events influence immune function. All of these must be taken on board. And yet, when we look at the data sets that we're interrogating, and here they are, summarized beautifully by Tim Radstacke and his colleagues recently, the data sets, the Human Microbiome Project, Genome Project, collect omics and some immune function, but almost no psychosocial measures. We're missing an enormous part of what all of us know is true, that medicine is art as well as science. And the final experiment for you before I finish. We went back to SERA, and with Gary McFarlane, we asked the question at baseline, what are the clinical and current immune factors like CRP, ESR, rheumatoid factor? What are the factors that predict poor functional outcome at 1 year? And the factors were HAQ score, HADS depression, HADS anxiety, employment, presenteeism and absenteeism, and body mass index. There wasn't an immunological factor amongst them. I was devastated. Gary McFarlane sat in triumph for at least a week. He was intolerable. I had to avoid Aberdeen. But actually, this scoring system works very well, and what it reminds us is that we're treating people, not molecules. So ladies and gentlemen, as I move, I hope, towards a molecular revolution, I want to remember the patients that sit at the center of it. This is a man in Glasgow. I've told the story often. For those who haven't heard it, it was a patient who came in who was the first patient I ever gave anti-IL-15 antibody to. He was in remission for a year afterwards, and one day came in and asked me why I had never interrogated him. Why did you never ask me why I helped you with your research, Prof? And the answer, of course, was I got informed consent. We do have IRBs, by the way, Len, just so you know. And but I didn't ask you why because I don't want to prejudice your decision. And he said, well, I'm going to tell you why. Big guy, Glasgow, going to tell you something, just say yes. I said, okay, tell me. And he put a photograph down on the table in the clinic room, and he put his hand next to it. And the photograph was of his firstborn grandson's hand. And he looked me in the eye and he just said, right, Prof, I want you to promise me that this, his hand, would never happen to that, his grandson's hand. I had to leave the room. That's what the patients want, ladies and gentlemen.